Yeah, thank you, everyone. Good afternoon. I'm glad to be here, and I'll be talking about biomarkers in precision medicine. Some of the biomarkers I've been studying in my career so far, and I'm glad to talk you through some of them, and then to welcome your questions afterward. So, all over the world, scientists have been interested in studying biomarkers, especially the one that can improve precision medicine. So biomarkers like diagnostic, monitoring, biosafety, susceptibility, and so on and so forth. And that is what I'm also interested in. And these biomarkers, because they are good in health and also in diseases, and these have been discussed in by various scientists. It could be gene-based, metabolism-based, source-based, protein-based, and as the case may be. So, but they must be specific, sensitive, and then have predictive values. So some of the biomarkers I've been studying, I started with monosodium glutamate. Actually, we eat it every day. We have eaten it this afternoon at lunch. So we found out that it elevates the level of AST, the enzyme in the liver, and ALT. And by targeting them, you can discover MSG toxicity. Then also pharmacodynamic biomarkers, I started exploring agents that have apigenidine, neotenine, and naringenin, and also magniferin. And we applied graded doses and then found out that they have antioxidant, anti-stress, and then anti-inflammatory properties, which are good. And furthermore, look at prognostic biomarkers like MDA, glutathione, SOD, catalase. These are enzymes that have been implicated in inflammation, and then also they have tissue necrosis effect that you could target when you are kind of interested in pathology. Then I move on to biosafety biomarker. So where we got interested in methyljasmonate, a compound that is similar to prostaglandins, and we found out that it decreases the corticosteroid level associated with anoxic stress to prevent early convulsion in people. Then I moved on to map all these biomarkers together, genetic biomarkers in particular, whereby we look at the complex biological functions in genetic biomarkers, which are mostly stress, neuroinflammation, apoptosis, mitophagy, and then also autophagy. But genetic disorders are very complex. And then in this first study of in silico analysis that I did with my colleagues, we mapped these pathways of genes that are implicated in precision medicine, and we found out that they have this unique arachidonic acid pathway, which is actually inflammation, and then is being linked to many diseases. And then in my PhD study, we picked up some courses, some patients, Parkinson's disease patients, and we found out a deletion in PAC7, which is actually a glutathione, and then a duplication of alpha-synuclein, which has been implicated in Parkinson's disease. These cases have been solved now. And then in the same project, we looked at so many patients from South Africa and Nigeria, and we identified rare genetic variant in 42 genes that are currently being explored. One of them is ATP13A2. Then when I started my first postdoc, I moved up to a lab where we were investigating, again, genetic biomarkers in hearing impairment, and we won a good grant for that. And in this study, we found out a mutation in DNA repair gene. This is a new identification in hearing impairment study, and it's being currently being studied. During that, we expanded our project to different African countries and also in the United States. And in Senegal, we explore a complex family whereby we found a mutation in GJB2, which is a gap junction gene that has been established in hearing impairment pathology. And this is interesting. So the more latest on MCPH1 that I talked about, the DNA repair gene, these studies are currently being reviewed in scientific report journal, whereby we also found a mapping of different location in the gene that different mutations have been identified. Last year, December, this study was published with my colleagues in South Africa, where we identified a triple 1B mutation in the Puticum family, whereby it's also a DNA repair gene implicated in cell cycle regulation. So currently, when I joined the University of Oxford, in Taylor's lab, Jimmy Taylor's lab, we started exploring different models that we can use. And one of the method that I came up with is to use multi-layer annotation to characterize genetic markers to underpin them. And now that is leading to a more advanced models like ranking of the genes, applying statistical significance, and then using data transfer in clinical settings. So this method was published last year, whereby we predicted the models that we can use in precision medicine. So using this model, we'll be able to apply to what I've done previously in hearing impairment, and currently with gestational diabetes, because I have a collaboration with somebody working on that, and then fibrosis, to identify novel genetic biomarkers and then prioritize this variant. And then once we do that, you cannot substitute in silico analysis or genetic bioinformatic analysis. We can't substitute with functional assay. So we have to do some functional assay, looking at the different mechanisms and pathological pathways that can they be predictive. So that is it. I would like to thank my funders, the universities that I've learned and currently working with, and then without them, I can't do anything interesting. Yes. Thank you very much.